been doing for the past year at CMS and what will hopefully become the first chapter of my dissertation. My story takes place in the Floridan Aquifer. As you can see, the Floridan Aquifer is a very large ecosystem that covers the state of Florida and even parts of Alabama and Georgia. It is very biodiverse and of high recreational value for the state, as well as economical value. It generates millions of dollars annually, so it's a very important ecosystem for us. However, as important as it is being threatened by environmental and anthropogenic stressors, such as the increase of nutrient loading in these areas. And one of the first indicators that is affected by these stressors are actually aquatic plants. Yes, plants are very cool. So the Florida springs have a variety of submerged aquatic vegetation and other type of photosynthetic organisms. They range from diatoms to something as complex as carnivorous plants, and they provide multiple services to the ecosystem, such as nutrient cycling, carbon sequestration, habitats and nesting areas for wildlife. And even though that they can have really good appliances for the ecosystem, under stress, they can become harmful, kind of like grad students during comps. And an example of this is the cyanobacterial mats increasing, or that they can be harmful for the ecosystem, blocking the sunlight to those submerged aquatic vegetation, and thus they're unable to carry out photosynthesis. And even though we have an idea on how they respond to stressors, their interactions with the microbiota is still unknown. And one of the most abundant microbiota in the world are viruses. And just to put it in perspective, if you go to the spring head, where you have the freshest and like new water being input to the spring, you can find around 10,000 viruses per mil in a single drop of water. And if we put it to perspective in the ocean, you can find around 10 million viruses per mil in a single drop of water. So yes, they are very abundant, and because they're obligate parasites, they require a host to replicate, and because of this, they're a bit hard to study. Luckily, I'm in a group of virus hunters, and in our lab, even though that it's largely unknown, like how viruses interact with plants as their hosts or infecting aquatic plants, we have developed different strategies to discover viruses in these type of organisms. Just last month, our lab, one of our lab's postdoc, discovered the first virus that infects seagrasses. The seagrass in general that was used was the Thalassia testudinum, or turtle grass, which is the most abundant seagrass in this region. Now, even though that this is a highly studied seagrass, it's the first virus that it's ever discovered that infects this plant. So that just puts you into perspective the little that we know about plant infecting viruses. And yes, now we have a virus that infects a marine aquatic vegetation, but what about the freshwater plants? And this is why I'm here. The main objective of this research is to identify viruses infecting submerged aquatic vegetation in the Florida springs. When I'm talking about submerged aquatic vegetation, I'm including diatoms, algae, terrestrial plants that are in kind of like in the water's terrestrial interface, and other vegetation associated. So not necessarily everything's a plant. We chose 4 springs to sample: the Volusia Blue Springs, Rainbow, Manatee, and Ichitucknee, all here with the stars and numbered here. From each spring, we collected around 87 individuals of submerged aquatic vegetation. And once we collected them, we did morphological identification, and we came up with 16 species in total for all of the springs. Down here, you can see just examples of some of the vegetation that we collected, such as dollarweed and Illinois pondweed. Once we collected those submerged aquatic vegetations, we proceeded to rinse them to remove any kind of epiphytes or bacteria, anything that would be on top in the surface of the plant, so we can actually have just plant material. Once we have that, we mush them up. It looks kind of like those like green shakes when you finish. And we have to purify the virus-like particles from the plant. So we mush them, we filtrate them through a 0.02 micron filter, and then subject them to a nuclease treatment, which is essentially treatment of enzymes to remove any nuclei that is not viral, because we really want to know what type of viruses are there. But we don't really know which viruses are there because there are not known viruses for these aquatic plants. After that, we proceeded to extract the RNA and DNA from this purification. And then we subjected them to what we call the sequence-independent single primer amplification, or CISPA. CISPA is a really cool method to use in this occasion because it amplifies random segments of unknown viral sequences, which helps us discover new viruses. And as I mentioned, viral hunters, job security. So when we have that CISPA products and we have those sequences, we clone them and then send them to sequencing. Thanks to Jeremy, now we know that's like the Google Translate. We get those different size sequences, and when we put them and compare them to the database, we're supposed to get the closest neighbor or closest related identity to what is in the database. But that, as Maya mentioned in Jeremy's talk, that doesn't necessarily mean that is that exact species. So with that said, in this pie chart, I wanted to show the diversity of the viruses recovered from the sequences that we obtained. I wanted to remind you that we were doing this on vegetation per se. So, and these are sequences, different size sequences that when we obtained them, these were the closest neighbors in the database. The highest diversity shown was for aquatic invertebrates, but aquatic invertebrate viruses. But as I mentioned, these came from plants. So how come they came back as sequences from aquatic viruses, aquatic invertebrates? So these are the closest neighbors. And that doesn't necessarily mean that they were from invertebrates. So I'm going to just, because I'm looking for viruses infecting plants, I'm just going to focus on the top portion when we had viruses that came back as for red algae, infecting diatoms, and infecting terrestrial plants. And as in the table, you have two columns. On the left, you have top hits of plant-infecting viruses, and on the right, you have the identity match to the database or the Google Translate. And for us, a good percentage of identity would be maybe 90 or above. But as you can see, these are low percentages. And if you're reading this table, you see that we can read potato virus and turnip virus, but these didn't come from a potato or a turnip. What is really the take-home message on this table is that these were the closest neighbors, and they came out as plant-infecting viruses, but they're not from aquatic plants, which means that there's hope for potential new viruses, because these were the sequences that were in the database, and we compared our sequences with them, and this is what we got back. So something similar, but not the same thing. So to solve this conundrum, I designed primers from sequences, the ones that we compared, which are kind of probes, to see if I can complete their viral genomes. And when we compared them to the database, hopefully they would be new viruses, right? That's perfect in the world of microbiology, right? Psych. What happened was that we got our sequences back, we did CISPA again, and we got the same values back. So the same lengths of the sequences and the same identity match. And what we also had was new viral hits. So we showed more viral diversity when we designed this new primer specific to those sequences. So even though CISPA did the part on doing amplification of random sequences, it really didn't help us to completing the genomes that we were looking for in these viruses. Which brings us to the next steps in this project, which I will be working this year. Instead of doing CISPA, we're going to do a broader or a faster way to do this is a high throughput sequencing. So instead of analyzing 5 sequences at a time. I'm going to analyze 5 million at a time. So we want to obtain the complete genomes and hopefully in the future just do some phylogenetics work to compare the viruses infecting terrestrial plants and aquatic plants. The submerged aquatic vegetation in Florida Springs is decaying at a fast rate. We don't really know what's the role that viruses are playing in their mortality, but hopefully by understanding this, we can better improve the health of the springs and maybe in the future improve management practices for the Florida Springs. Thank you.